So chapter 15 is now learning about the chromosomes and how they actually work to match this genetics we are learning about in chapter 14. So the first thing is that before we really knew anything about chromosomes, we've been learning about alleles and Mendel's ideas and Punnett squares and all of that, but the only thing they called them were hereditary factors. They didn't really call them genes. They didn't know anything of that. They were just something that carried information. And now we know that they're located on the chromosomes, but we didn't know that before. And so now that once scientists started discovering that they're all on chromosomes, it became much easier, and a lot of patterns were explained. Okay, so mitosis and meiosis were first discovered in the late 1800s. Mitosis first about 1875, meiosis about 1890. And once scientists started understanding the actual process of meiosis and looking at the chromosomes, they started realizing, okay, this really matches with Mendel's ideas of heredity. They saw a ton of parallels. For example, they saw that that both chromosomes and genes are present in pairs, and Mendel knew that, and they saw the pairs of chromosomes during mitosis and meiosis, and they saw that homologous pairs and alleles separate during meiosis, and that they reunite during fertilization. So they're like, that's pretty crazy that those would both happen. They must be related. And so once that happened, they came up with the chromosome theory of inheritance, and that says that all these Mendelian genes or hereditary factors that we've been learning about, they have specific locations, loci, on chromosomes. So the gene for pea color is found on one of those pea plant's chromosomes. And we also know that these chromosomes undergo segregation and independent assortment. And so the act of the homologous pairs assorting independently explains a lot of things like Mendel's law of segregation, how different genes are not inherited together, because different homologous pairs are not necessarily inherited together, and independent assortment, because the two of those things go together. So, okay, this is a really great figure on page 287 of your textbook, and it shows a parent that's homozygous dominant for both traits and a parent that's homozygous recessive for both traits. And they're only going to make one kind of gamete, right? Because if you FOIL to find the gametes, then this guy can only give big R and a big Y, so that's the only kind of gamete he's going to make. And this guy can only give big Y and big R, so that's the only kind of gamete he's going to make. And so then we see that when these two gametes unite, we're going to have a heterozygous. Capital R, lowercase r, capital Y, lowercase y. That's what we have. So that's our F1 generation. They're all going to be round and yellow. When we do another cross of the F1 generation, an F1 cross, we would expect to find 9331. That's what Mendel found. And when they actually look at the chromosomes, you can see why. So the same thing happens again. We can make four different kinds of gametes. And so you see that a quarter of the gametes are gonna be big Y, big R. A quarter are going to be little y, little r. A quarter are going to be big Y, big R, and a quarter are going to be little y, big R. And so that would give us the same outcome. So you can, by looking at the chromosomes, you can see that it matches perfectly with what Mendel thought. So it's pretty crazy that Mendel had it right all this time when he didn't even know anything about these two things. And so we can see from the law of segregation says that the two alleles for each gene separate during gamete formation. So that happens during anaphase 1. We know that. We separate the homologous pairs, we separate the alleles. The law of independent assortment says that the homologous pairs assort differently from each other. So you can see here, the parental and maternal chromosomes stay together, whereas here they separate. So that's independent assortment. It's totally random which genes from which parent the gamete are going to end up with. Okay, so after we learned all this, there was a scientist named Thomas Hunt Morgan, and he did an experiment using fruit flies, which really helped us understand and kind of prove that the chromosome theory of inheritance. So he did an experiment with these fruit flies, and what he did was he bred them for basically about 2 years, studied them under a tiny little microscope, but then he realized nothing was really happening, and he was bummed that after 2 years, there wasn't anything going on. One step backwards, the reasons why fruit flies are important is because they produce tons of offspring. Like one mating gives off 100 offspring, or hundreds of offspring. And they can produce every 2 weeks. So that's pretty ideal to study them because you don't have to wait a long time to see the outcomes of your crosses. And they only have 4 pairs of chromosomes. So it's pretty simple to break things down and figure out where things are coming from, as opposed to humans where our generation time is 30 years instead of 2 weeks. You produce 1 offspring, maybe 2, if you're lucky. More than that's very rare per mating, and we have 23 pairs of chromosomes. So obviously fruit flies are a pretty good subject of study. And so what he did was he's watching them for years, nothing's happening, and then finally he sees one male that has white eyes instead of red eyes. So every other fruit fly for 2 years had red eyes, and then suddenly one has white eyes. And so he realizes that this must be a mutation that happened. And so he starts identifying the difference between mutants and wild type. That's what we call the normal ones, the unmutated ones. So the wild type had red eyes, the mutant had white eyes. And so then he starts doing more tests, crossing the white, he crosses the white-eyed male fly with a female red-eyed fly. And he sees that all F1 generation have red eyes, which you would probably expect. The female is probably homozygous dominant, and the male is probably homozygous recessive. And so that's fine. But then what he sees from the next generation, so the F1 generation, they all have red eyes. The next generation, he sees 3/4 red, 1/4 white. Again, what we would expect. Given an F2 cross, that 1/4 of them would be homozygous recessive. And so that was fine. But then he realizes that all of the females still have red eyes, and half of the males have white eyes. So it's not, there's a differentiation between genders, which is not what he was expecting. If it had been normal, it would have been 3/4 of females have red, 1/4 have white, 3/4 of males have red, 1/4 have white. Instead, it was all females have red, half the males have red, half the males have white. So this is the first experience of a sex-linked trait. And he's able to determine from this that traits are carried on chromosomes and that this trait must be carried on the X chromosome. Because it's the only reason to account for why males end up this way as opposed to females. So that supports the chromosome theory of inheritance because they must be located on the chromosomes. And that is all for chapter 15.1.